So today we are going to discuss about monoclonal antibodies. So first we have to know what is an antibody. Generally, when the antigen enters into our body or a foreign substance or antigen enters into our body, within our body we have antibodies which will fight with that antigen and which will stop the activity of that particular antigen. That is nothing but we have immunity because of the presence of antibodies within our body. So antigens contains epitope which is present on the external surface of the antigen, whereas an antibody contains paratope. This epitope and paratope will combine so that we will have antigen antibody reactions. So as we have discussed in the previous slide, antibody is nothing but which is present within our body. It will recognize whatever the foreign substance, virus or bacteria that enters into our body and fight against that particular antigen. So it is called as antibody. So we are terming the next term monoclonal antibodies. Mono indicates single. So monoclonal, which are cloned from a single parent cell. So they are called as monoclonal antibodies. Whereas the next term is polyclonal. Poly means many. So polyclonal antibodies are prepared from different cell lines. So they are called as polyclonal. And the next one is immunoglobulins. These immunoglobulins, we have a structure similar to that of a glycoprotein and they also act as antibodies. We have seen that antibodies are present within the body, whereas monoclonal antibodies are prepared from the single parent and polyclonal from the multiple cell lines. So who started this preparation? Neil George Kohler are the people who started the principle and production of these monoclonal body antibodies, and they have been won the Nobel Prize in the year of 1984. So in the year of 1986, the first monoclonal antibody reached the market, and the drug particular monoclonal antibody is muromonab. Whereas first they are prepared from the animal, mouse. Then they started preparing completely from the human beings. In the year of 2003, completely human monoclonal antibody is released, that is adalimumab. So we started preparing these monoclonal antibodies. So why they have prepared? What are the advantages of these monoclonal antibodies? Homogeneity. Nothing but how many ever antibodies you are producing, they have a similar action and they specifically act on the certain antigen. They have a specificity to select the particular antigen, and we can produce numerous amount of the antibodies. How much ever quantity you want, you can produce. There are certain disadvantages also for this monoclonal antibodies. That is, the capacity to bind with the antigen will be less when compared to the polyclonal antibodies. And if the conformational change, the structure is changed of antigen, then they can't act on that particular antigen. So specificity is not there. And sometimes what happens is it acts on some other different antigen. So cross reactions may also happen, and the time for preparing these monoclonal antibodies is very large. So as we started preparing these monoclonal antibodies, what is the purpose? What are the applications of these monoclonal antibodies? They can be used for diagnostic testing to check certain disease is present or not, whether pregnancy is present or not. We can detect whether the cancer is present or not. If the cancer is present, we can start giving radioimmunotherapy for the cancer. We can treat the viral diseases. We can treat the cancer through the drugs. And the pathogens, presence of pathogens can be detected. The organ rejection can be minimized, and rhesus disease immunization. This rhesus disease is nothing but the antibodies of a mother will start acting on the baby's blood cells. This is called as rhesus disease, so it can be immunized. So production of monoclonal antibodies. Generally, when an antigen or a foreign substance enters into our body produces antibodies to fight against them. So here we use the same formula. What we will do is we will immunize the mice. The antigen will be injected into the mice. So what the mice will do? It will start producing the antibodies. So what we will do is the splenocytes which are producing the antibodies are separated, and myeloma cells. These myeloma cells, they are immortal. Immortal. Mortal means death. Immortal means they don't have the death and they can rapidly divide. So what we will do is we will do the fusion of these splenocytes and the myeloma cells. So, and then we will check whether our antibodies which are produced from these splenocytes are active against the antigens. So, how we will check this? Functional characterization. By using the ELISA method, we will check whether our antibodies are having the activity against the antigens. If once they have proved that our antibodies are active against the antigens, then we will start cloning our antibodies and we will expand the clones for the production of the antibodies. This is how the monoclonal antibodies are produced. Yes. So evolution of monoclonal antibodies. In the initial stages, we have prepared the antibodies completely from the mouse proteins. That is 100% mouse, which are called as first murine or first generation monoclonal antibodies. Then in the later stages, what we did is we have taken certain quantity of antibodies from the mouse and chimeric. Chimeric means they are prepared from different various parts of various animals. Then the third humanized, that is 10% we have been taking the proteins from the mouse and remaining from the humans, that is third humanized. And the last one, the improvised version, is fully human. The antibodies are completely produced from the human. So what is the mechanism of these monoclonal antibodies? How they are acting in our body? So what they will do is they act on the cancer cells. They will trigger the destruction of the cell membrane. The cancer cell membranes are destructed and the growth of the cancer cell is being prevented and the blood vessel growth is also prevented. And they inhibit or block the action of immune cell inhibitors. The immune cell inhibitors are the one which are inhibiting the activity of our immune system. So that particular inhibitors are being blocked. They directly act on the cancer cells and they help for the radiation treatment. Nothing but other healthy cells are not being affected with this radiation. Only cancerous cells have been affected. They bind with the cancer cells and immune cells and thereby they help for the treatment of the most of the cancers. Coming to the pharmacokinetics of this monoclonal antibodies. Most of the monoclonal antibodies are preferred to be given by IV route so that they will have 100% bioavailability. The volume of distribution for these monoclonal antibodies will be small and the tissue penetration is also less. And depending upon the nature of the antibody, they can remain in our circulation from 2 hours to 2 weeks. And all the monoclonal antibodies, they are very expensive and they are eliminated and uptaken by the catabolism by reticuloendothelial cells and the target tissues. Coming to the side effects of these monoclonal antibodies, they may produce allergic reactions like hives, nothing but a red spot or swelling, itching, flu-like symptoms, which includes the chills, fatigue, fever, muzzy aches and pains. And the most common symptom, nausea, diarrhea and skin rashes. Coming to the nomenclature of the monoclonal antibodies, the prefix, it can be varied from drug to drug, and the target infix. This is from where it is prepared. What is the target? The source and the target of that particular drug gives the target infix and source infix. So the target is bone. So we will write it as O. O indicates osteo, that is bone. V for viral, B for bacteria, L for immunomodulating, C for cardiovascular. Like that, depending upon our target, we are going to give a target infix. And coming to the source infix, from where it is prepared, whether from the human, we give it as U, mouse, we give it as O, hamster, E. So like that, whether it is humanized, ZU. So according to the source, we will add the source infix. And most of the drugs will have a suffix which is MAB. So here are certain examples for the monoclonal antibodies with their indication.